Hi, my name is Hamna Salimi,
And I'm Linda.
and this year our project was analyzing water bodies and their microplastic quantities.
The purpose of this project was to gain a better understanding in the amount of microplastics present in the different bodies of water. To do so, we collected water samples from different water bodies and analyzed the quantifiable amount of microplastics.
Background research. The most prevalent type of marine debris found in our oceans are plastics. These plastics that end up in our oceans never really go away as they are constantly breaking down. Plastics that are less than 5 millimeters in length are known as microplastics. Microplastics are a fairly new and emerging field of study as there is not much known about them. The first step to understand their impact on the environment is understanding how much gets released into our oceans each year. Limited surveys show that children and adults ingest anywhere from 100,000 microplastic specks per day. Microplastics are a growing concern as plastic productions and waste continue to emerge. Microplastics are so small that they can even enter our cells and tissues, which becomes a concern as they are now a foreign particle substance which is present in our body. This can lead to other issues such as inflammation of lung tissue, which could eventually lead to cancer. Microplastics are also harmful because of the toxic chemicals that are present in them. These toxic chemicals may come into the microplastics either from the original site of plastic production, or microplastics may absorb chemicals from their environment later on. For example, polychlorinated biphenyls, or PCBs, are a toxic chemical which are directly related to cancer and other health concerns. PCBs are present in many microplastics. Microplastics may come from a variety of sources including microbeads, which may come from single-use plastic items such as plastic bags or tire wear, and even personal care products. Additionally, in these oceans, microplastics are very hard to track because of their small size and the large size of the ocean, but also because oceans have different current circulation patterns, which makes it hard for them to track if they are constantly being moved around.
So for the hypothesis, we assume that as the water bodies get closer to industrial sources and residential areas, the amount of microplastics will be increasing in the 500 milliliter water sample collected. This is due to the fact that in those sources and areas, there are direct pollutants such as tire wears, plastic bottles, and plastic bags. Microplastics may also make their way into those big water bodies such as lakes and oceans through the small ones, as we mentioned, ponds and creeks. Additionally, as the amount of microplastics increases, the pH level will also increase, but not necessarily in a linear relationship. This is because there are many factors to the high pH level, but microplastics are only one of them. And it is not reasonable to say that it will increase in a linear pattern. The materials we use for the experiments are 500 milliliter water bottles, pocket microscopes, and the fine mesh net. Our independent variable was the water bodies, and the dependent variable was the microplastics per 500 milliliter bottles of water. Our control variables was the amount of water collected and the weather, time, and date the water was collected, and the type of mesh net used.
For our procedure, the first thing we did was collect 500 milliliter samples of water from the different water bodies. We made sure to collect 3 500 milliliter samples so that we had 3 trials and we can better calculate a more accurate average. Additionally, we had a control group in this experiment which was tap water, which is assumed to have 0 microplastics. We also made some qualitative observations while we collected our water bodies, such as the water body movements on some beaches. After we collected our samples, we had a fine mesh net strainer and we ran our samples through that mesh net strainer. Once we ran it through, there were some particles and microplastics collected onto the strainer, but to make them more visible and clear, we decided to make an imprint from the strainer to a white napkin, and we pressed really hard so that all the particles from the strainer move onto the napkin. Once those particles had moved onto the white napkin, we used our pocket microscope and put it onto the napkin and count how many microplastics we saw. We assumed that the multicolored and string-like particles were microplastics, and we counted them the best we could. Once we had counted them, we recorded these numbers and we calculated the average from our 3 trials. The last thing we did was also measure for some chemical qualities such as pH, ppm total alkalinity, ppm total stabilizer, and ppm-free chlorine to see if there was any relationship between our chemical properties as well as the water bodies.
In conclusion, as seen in Figure 1, the water body with the most amount of microplastic was Milestone Creek, while the amount with the least microplastic was Lakeside Park, which is southwest Lake Ontario. Looking at the pH level graph, there is not a distinct relationship between the levels and the amount of microplastics.
Analysis of conclusion. So our conclusion supports the original hypothesis in which a creek had the highest amount of microplastics. Because the creek is located near residential buildings, there are more microplastics entering directly into the stream, such as different tire wear and single-use plastic items and other pollutants. The creek that was analyzed in this project was located right beside a park, which could indicate a lot of daily activity going on in the park, which can also contribute to more pollutants. As compared to our Lake Ontario, on the other hand, we collected different parts of Lake Ontario, and each of these parts had some water movements going on, some more than others. This is because this was a beach, and when we went to that beach, there was current and wave movement which was moving around the microplastics, which could be why there was fewer in Lake Ontario as compared to the creek where there's very still water movement. Additionally, when we went to the Lake Ontario area, there was some rocks on the beach which acted as a natural filter for the microplastics. However, it is very likely that there could have been some nanoplastics in these Lake Ontario samples, as we did not use a high accuracy microscope for our experiment. Some future steps for this project could be to measure additional ocean and lake sources and measure their microplastic quantities than the ones we used in our study. Another thing that we could look into is comparing current and wave movements to the amount of microplastics present and see if the amount of wave movements going on at a certain body of water also impacts the amount of microplastics we may find.
So one of the challenges we faced during the experiment was finding a good day to collect the water samples. We didn't want to collect the water too late in the winter since the lakes and ponds might have been frozen. Frozen lakes and ponds stop water from circulation, which might affect the amount of microplastics collected. It also traps the microplastics in the ice, which could lead to less microplastics than expected.
Additionally, another challenge that we experienced was trying to come up with a high accuracy procedure. Originally, when we collected our water samples, we did not have a microscope and we were finding little to no microplastics in all of the water bodies. We then collected our samples again and we analyzed them again, this time using a microscope, which led to more accurate microplastic quantities being found in each of our water bodies.
We'd like to thank our parents, STEM leaders, and Mr. Sanderson for supporting us.
And thank you so much for listening to our project. We hope you enjoyed listening to it as much as we liked doing it.
Thank you.